Hello there, and welcome to the Sleepy Science Secrets. Tonight, we are going to wander very gently into one of the softest mysteries the natural world has ever kept. The secret mathematics hidden inside every flower that has ever bloomed. Imagine this: every petal, every curve, every tiny thread of color that you have ever admired in a garden or a field, or a single blossom sitting in a glass of water on a windowsill. All of it is quietly following a pattern so ancient and so precise that mathematicians have given it a name. And that name is beautiful. Flowers do not simply grow, they count, they arrange. They fold themselves along invisible lines of balance, and the result is something so graceful that humans have been pressing flowers into books and painting them onto walls for thousands of years, not fully knowing that what moved them so deeply was the hidden geometry inside. Tonight, we will find it together, slowly, softly, one petal at a time. Let yourself settle now. Feel the surface beneath you, how it holds you without effort, how you don't have to do anything at all to stay right where you are. Let your breathing slow just a little. Not forced, not controlled, just easy. And imagine, if you like, that you are lying in a very soft meadow, and above you the sky is turning that particular shade of blue that comes just before the stars appear. Around you, very gently, flowers are closing for the night. And as they close, we are going to look at them very closely, not in a hurried way, but in the way that a bee looks at a flower, hovering still, taking in every detail with complete and unhurried attention. The first thing to understand about flower symmetry is something called radial symmetry, and it is perhaps the most peaceful of all the shapes that nature makes. A flower with radial symmetry looks the same from any direction you choose to look at it. Turn it one way, and it is balanced. Turn it another, and it is still balanced. Imagine a daisy, the kind you might find growing in a path's edge, its white petals fanned out around a golden center. From above, it looks like a tiny sun. From any angle around its face, it is exactly the same. This is radial symmetry, and it has another name that scientists use. They call it actinomorphic, a word that comes from the Greek for ray and form. Ray formed, a flower shaped like a ray of light from a center. You can let that word drift through your mind without needing to hold onto it. What matters is the feeling of it, the evenness, the roundness, the way such a flower spreads its welcome equally in every direction, as if it has no preference for who approaches. And most flowers do this. Most flowers are, at their heart, radially symmetrical. The wild rose, the buttercup, the magnolia, the cherry blossom, all of them built around a center, all of them spreading outward in even, repeating measures. There is something in seeing this that settles something in the human chest. We are drawn to symmetry, to evenness, to the sense that a thing is balanced and whole. Scientists who study human perception have found that we recognize symmetrical faces and symmetrical flowers more quickly than asymmetrical ones, and that the recognition itself is accompanied by something like a small, quiet pleasure, not an excitement, just a rightness, a sense of things being as they should be. Now, within that radial symmetry, flowers do something even more interesting. They choose a number. Each flower seems to decide, very early in its development, how many petals it will carry. And these numbers are not random. They follow a pattern so beautiful and so widely distributed through the natural world that it has become one of the most famous sequences in all of mathematics. You may have heard of it. It is called the Fibonacci sequence. It begins like this: 1, 1, 2, 3, 5, 8, 13, 21, 34. Each number is the sum of the two before it. 1 plus 1 is 2, 2 plus 1 is 3, 3 plus 2 is 5, and so on quietly and endlessly, each number growing out of the ones that came before it, like a plant growing out of the soil that holds its roots. Buttercups have 5 petals. Look at any buttercup and count them. You will find 5, almost always. Bloodroot, a soft white wildflower that blooms in early spring in North American woodlands, has 8 petals. Delphiniums often carry 13. Some asters have 21 petals. Some daisies have 34. Others have 55. And there are sunflowers, we will come to sunflowers, that carry 89 petals around their great golden faces. 5, 8, 13, 21, 34, 55, 89. The Fibonacci sequence, written in petals on the living faces of flowers. This is not something that flowers have been taught. No one arranged them this way. It emerges from the physics and chemistry of how plants grow, from the way that new petals push out from the growing tip of a plant, each one forming at a precise angle from the one before it. The mathematics is built into the growing itself. That angle, the angle at which each new petal or leaf or seed forms relative to the last, is approximately 137.5 degrees. It is called the golden angle, and it is derived from a deeper mathematical relationship called the golden ratio. The golden ratio is roughly 1.618. If you divide any Fibonacci number by the one before it, say 34 divided by 21, you get something very close to 1.618. The further along the sequence you go, the closer you get. The golden ratio and the Fibonacci sequence are, in a way, the same thing seen from two different directions. And together, they are the hidden architecture of flowers. You don't need to remember the numbers. Just feel the idea. There is a ratio, a relationship, a proportion that runs through the natural world like a quiet thread. And flowers have been following it for hundreds of millions of years without knowing it, simply because it is the most efficient way to grow. Now, shift your attention very gently to a sunflower. Imagine you are looking at one, a large one, with its great circular face tilted slightly toward you in the late afternoon light. The outer petals are bright yellow, glowing almost, and the center is a dense, intricate spiral of seeds. Look at those seeds. If you look carefully, you can see two sets of spirals, one set curling to the left, one set curling to the right. The spirals going one way and the spirals going the other way are almost always Fibonacci numbers. A sunflower might have 34 spirals curling clockwise and 55 curling counterclockwise, or 55 one way and 89 the other, or 89 and 144, always Fibonacci numbers, always two consecutive numbers from the sequence, nested inside each other, braided together across the face of a single flower. This arrangement is not decoration. It is engineering. It is the most efficient way to pack seeds into a circle so that no space is wasted and every seed has access to sunlight. The flower found the answer to an optimization problem that human engineers only recognized and named in the 20th century. It found it through millions of years of gentle refinement, each generation pressing a little more efficiency into the pattern until the pattern was very nearly perfect. And this arrangement has a name. It is called phyllotaxis, from the Greek for leaf arrangement. Phyllotaxis is the study of how plants arrange their parts, their leaves, their petals, their seeds, their branches, in space. And what phyllotaxis has found over centuries of careful observation is that plants are astonishingly consistent. They do not vary wildly. They follow the same angles, the same sequences, the same proportions, across species that have never shared an ancestor, across plants that evolved on different continents, across flowers that live in deserts, and flowers that live in rainforests, and flowers that live at the edge of glaciers. The mathematics is the same. The golden angle is the same. The Fibonacci numbers appear again and again and again, as if the universe itself has a preference for this particular way of arranging things in a circle. Let your eyes grow a little heavier now, if they would like to. There is no hurry here. We are not going anywhere. We are just sitting with the flowers in the soft light, counting petals and spirals, and noticing the way the world has been arranged with such extraordinary, quiet care. And if you're enjoying this gentle journey through the science of flowers, feel free to leave a little comment below. It means so much to know that you're here, resting along with us. And if you haven't already, there's a whole playlist of these slow, sleepy explorations waiting for you when you're ready. Now, let us move from the Fibonacci numbers to something slightly different, a different kind of symmetry that some flowers have chosen. While most flowers are radially symmetrical, some flowers are bilaterally symmetrical. A bilaterally symmetrical flower has exactly one line of symmetry. One line you can draw through it that divides it into two matching halves, like a mirror placed vertically down the center of a face. These flowers look the same on the left as they do on the right, but they are not the same from every direction. They have a front and a back, a top and a bottom. Scientists call this kind of symmetry zygomorphic, another beautiful Greek word, meaning yoke-shaped, as if the two halves of the flower are joined like a pair of animals yoked together for the same purpose. The orchid is perhaps the most famous of the bilaterally symmetrical flowers. Orchids are extraordinary creatures. There are over 25,000 species of them, making them one of the largest plant families on Earth, and almost all of them are bilaterally symmetrical. You can draw one line, and only one line, through an orchid blossom that divides it into two equal halves. This bilateral symmetry is not an accident. It is a conversation with pollinators. A bilaterally symmetrical flower has a specific orientation. It opens in a specific direction. It presents its landing platform, its nectar, its pollen in a specific arrangement. An insect approaching from the front experiences the flower very differently from an insect approaching from the side. And this directionality means that the pollinator, usually a bee or a butterfly or a moth, approaches from the same angle every time, picks up pollen from the same place every time, and deposits it on the next flower in the same place every time. The bilateral symmetry of the flower is a landing instruction, a guidance system, a very gentle and very beautiful form of communication. Snapdragons are bilaterally symmetrical. So are foxgloves, with their tall spires of tubular bells, each one patterned with spots inside to guide a bee toward the nectar. So are sweet peas and violets and irises and the delicate wildflower called the toadflax. All of them have that single line of mirror symmetry, that one fold along which they would match perfectly if you could fold the air itself. And in each case, the symmetry is in service of a relationship, the flower and the pollinator, moving through the world together, each one shaped over millions of years to fit the other. This coevolution between flowers and their pollinators has produced some of the most extraordinary shapes in the natural world. There is an orchid in Madagascar called Darwin's orchid, or the star orchid, whose nectar is stored at the bottom of a tube nearly 30 centimeters long. When Charles Darwin first saw it in 1862, he predicted that there must exist a moth with a tongue exactly long enough to reach that nectar. Everyone who heard this thought it was a rather extraordinary claim. 41 years later, in 1903, the moth was found. It was a species of hawk moth with a proboscis, a feeding tube, almost exactly 30 centimeters long. The flower and the moth had shaped each other across millions of years, each one pulling the other toward a more and more extreme and more precise relationship, until the flower's tube and the moth's tongue were matched like a key and a lock. The bilateral symmetry of the orchid, the precise geometry of its opening, the exact angle at which it presents itself, all of it part of this ancient, ongoing conversation. And now, while we rest in that thought, the idea of a flower and a moth finding each other across millions of years of patient adjustment, let us turn to something even more intimate. Let us turn to the inside of a flower, to the parts that most of us rarely look at closely, and find the symmetry there as well. Inside most flowers, there are two sets of reproductive structures: the stamens, which produce pollen, and the pistil, or pistils, which receive it. And these structures are almost always arranged with symmetry too. In a radially symmetrical flower, the stamens often appear in whorls, rings arranged around the central pistil, each stamen at an equal angle from its neighbors, so that the whole interior of the flower looks like a tiny wheel of spokes. The number of stamens often, though not always, relates to the number of petals. A flower with 5 petals might have 5 stamens, or 10, a multiple of 5. The symmetry of the outside is echoed in the symmetry of the inside, as if the flower is consistent all the way through, as if the balance is not just a surface feature but a deeply held principle. In some flowers, this internal symmetry is extraordinarily precise. The passion flower, which grows wild across South America and is cultivated in gardens around the world, has an interior so geometrically complex that it looks like something drawn with a compass. There are 5 sepals and 5 petals, arranged alternately so that no two of the same kind are adjacent. Then there is a corona, a ring of filaments, thin and colorful, arranged in a perfect circle. Then 5 stamens and 3 styles, the stalks that carry the pollen receptors. The whole flower looks like a mandala. It looks like something made with deliberate and careful human intent. And yet it grows this way because of the physics of plant development, the same mathematical pressures that produce Fibonacci numbers in seeds and golden angles in petals. The passion flower is a kind of natural diagram a flower that shows its mathematics more openly than most, wearing its geometry on the outside like an illuminated manuscript. You might, if you are comfortable, let your body sink a little more deeply into whatever is holding you right now. Let your shoulders be heavy. Let your hands be open or loosely curled, whichever way they have found to rest. There is nothing that needs doing. We are just here in the soft company of flowers and mathematics, letting the ideas drift by like clouds on a slow afternoon. Let us speak for a moment about color and symmetry, because they are more connected than you might expect. Many flowers have markings, lines, spots, gradients of color that are arranged with the same symmetry as the petals themselves. These markings are called nectar guides, and they function like the runway lights at an airport, directing pollinators toward the flower's nectar and pollen. In a radially symmetrical flower, the nectar guides often radiate outward from the center in lines that follow the symmetry of the flower itself. Equal lines at equal angles, like the spokes of a wheel, all pointing toward the same central source. In a bilaterally symmetrical flower, like a violet or a foxglove, the guides are arranged along the axis of symmetry, drawing the eye and the bee toward the center and inward. But here is one of the most remarkable things about these color patterns. Many of them are invisible to us. The human eye sees light in a range of wavelengths from red to violet, what we call visible light. But bees can see a broader range. They can see ultraviolet light, wavelengths that are entirely invisible to us, that lie beyond the violet end of our spectrum in a region we cannot perceive at all. And many flowers have patterns in ultraviolet light that are completely hidden from human eyes. A buttercup that looks like a uniform, glossy yellow to you appears, to a bee, to have a dark center, a bullseye, a target. The yellow petals absorb ultraviolet light toward their bases, creating a dark region around the reproductive center, while the tips of the petals reflect ultraviolet light and appear bright. The bee sees a flower that is not a simple yellow disc, but a complex, precisely arranged pattern pointing directly to the nectar. Scientists discovered these ultraviolet patterns by photographing flowers through special cameras equipped with ultraviolet sensitive film. The photographs revealed a hidden world. Flowers that had seemed simple and uniform, suddenly showing concentric rings, radial lines, complex spotted patterns, all arranged with the same symmetry as the visible flower, all invisible to human eyes, all perfectly legible to the bees that read them every day. It was as if the flowers had a second language, one they had been speaking for millions of years, perfectly audible to their intended audience, completely silent to ours. And so flowers carry this hidden symmetry, this ultraviolet language, as naturally as they carry their visible color and scent. And we might wonder, how many other hidden symmetries exist in the natural world? How many patterns have been arranged with such care and such precision in wavelengths and frequencies that our senses were simply never designed to perceive? It is a thought that could be exciting or overwhelming depending on the moment. But tonight, softly, it feels more like a comfort, like knowing that the world is richer than it seems, like knowing that there is always more gentleness, more order, more quiet care embedded in the fabric of things than any one pair of eyes can take in. Let us now think about the petals themselves, not just their number or their arrangement, but their shape. Each petal is a marvel of biological engineering, and that engineering is, again, deeply symmetrical. A petal is usually symmetric along its own long axis. The left side mirrors the right. This individual petal symmetry means that even when you look at a single petal, removed from the flower and held up to the light, you are looking at something balanced and whole in itself. The veins of the petal, the thin lines of tissue that carry water and nutrients from the base of the petal to its tip and edges, branch in patterns that mirror each other on either side of that central axis. The edges of the petal, whether smooth and simple, or scalloped, or fringed with tiny teeth like the petals of a carnation, repeat themselves from left to right. This symmetry of individual petals develops through a process called mirror imaging in the growing tissue. As a petal grows from its base outward, the cells on either side of its central vein receive the same chemical signals at the same time, and they grow in response to those signals in matching ways. The same hormones, the same quantities, the same timing. And so, the same shape emerges on both sides simultaneously, as if two artists working from opposite ends of a canvas are producing the same drawing. Scientists have been able to disrupt this process experimentally, producing asymmetric petals by interfering with the chemical signals, and the resulting flowers are immediately recognizable as wrong, not dangerous, not harmful, just clearly out of order, clearly not what the flower intended, in a way that we recognize almost before we have consciously seen it. The shape of a petal is also determined, in part, by mathematics at the cellular level. Each cell in a petal has a certain stiffness and a certain tendency to expand. The pattern of expansion, which cells expand more and which expand less, and in which direction, determines the final curve of the petal. Mathematicians who study this call it differential growth, and they have found that the complex, fluted, cup-shaped forms of flowers like roses and poppies arise naturally from relatively simple rules about which parts of the petal expand more than others. A flat sheet of tissue, given the right instructions about where to grow faster and where to grow slower, will naturally curl and cup and fold into a three-dimensional form, the way a wet piece of paper curls as it dries unevenly. The beauty of a rose petal, its softly curving edges, its gentle inner cup, the way it folds around the other petals before the blossom opens, is all the result of mathematically simple but spatially complex patterns of differential growth. The flower computes its own form, cell by cell, using rules simple enough to be encoded in a few molecules. And the result is something that has moved human beings to poetry and painting and tenderness for as long as we have been aware of flowers at all. And if you have a moment, and if this feels right, do leave us a like on the video. It genuinely helps this little channel grow the way a flower grows toward light, quietly and without hurry. And if you haven't explored the playlist yet, there are many more of these gentle journeys waiting for you there, each one a different door into the soft wonders of the world. Let us wander now into the world of spirals. We touched on the spirals of sunflower seeds, but spirals appear in flowers in other ways too, and each time they appear, they carry that same quiet quality of mathematical intention. The unopened bud of a rose is perhaps the most beautiful spiral in the common world. If you could look down into a rosebud from directly above, before it has begun to open, you would see the petals arranged in a perfect logarithmic spiral, the same mathematical curve that appears in seashells, in the arms of galaxies, in the arrangement of seeds in a pine cone. The petals are nested inside each other, each one at that golden angle of 137.5 degrees relative to the one before it, wound around the center of the bud in a spiral that has no beginning and no end, no corner and no straight line, only the continuous, unbroken curve of something turning toward the light. The logarithmic spiral is special among spirals because it keeps the same shape as it grows. A circle that grows outward while also rotating will either spiral in a way that changes shape, getting more elongated or more compressed, or it will trace a logarithmic spiral in which every turn is geometrically similar to every other turn. The spiral of a nautilus shell looks the same whether you are looking at a small nautilus or a large one, because the ratio of each spiral arm to the previous one is always the same. And the spiral of a rosebud has this same self-similar quality. As the rose opens and the petals unfurl, the spiral does not distort or change character. It simply expands gracefully and continuously, maintaining its proportions all the way from the first tiny loosening of the innermost petal to the full, open bloom. There is a word for this self-similarity. It is the word fractal, though fractals, properly speaking, are slightly different things, patterns that repeat at every scale. But the quality of self-similarity in natural spirals, the way they look the same close up and far away, the way the part echoes the whole, is something that mathematicians and artists have found endlessly fascinating. The Fibonacci spirals in sunflowers, the logarithmic spiral in a rosebud, the similar spirals in pine cones and cacti, and the arrangement of leaves on a stem. All of them instances of this same underlying tendency in nature toward forms that are consistent at every scale, toward shapes that replicate themselves inward and outward in the same proportions. Let your breathing be gentle and even now. Let each breath be as unhurried as a flower opening. You don't need to be anywhere. You don't need to do anything. You are just here in the soft company of mathematics and petals and spirals, and everything is very, very quiet. We should speak now of the violet. The violet is a small and very common flower found in hedgerows and woodland edges across much of the northern hemisphere, and it is a flower with a secret. The violet produces two kinds of flowers. The first kind are the ones you can see, the familiar purple or white flowers, bilaterally symmetrical, opening in spring, fragrant, visited by bees and other pollinators. But there is a second kind of violet flower, one that you would almost certainly never notice unless you were looking for it specifically, because these second flowers never open. They remain closed, tightly furled, pressed close against the soil under the leaf litter. They are called cleistogamous flowers, from the Greek for closed marriage, and they fertilize themselves from within without ever opening, without ever offering themselves to a pollinator. The pollen never leaves the anther. It grows through the anther wall, directly into the ovary of the same flower, fertilizing the seeds without any outside assistance at all. These hidden flowers are not symmetrical in the way the open flowers are. They are simple, reduced, without the elaborate bilateral symmetry of the visible spring flowers. But they serve a different purpose. They are insurance. In a bad spring, when pollinators are scarce or the weather prevents the visible flowers from being visited, the hidden flowers ensure that the violet will still produce seeds. The plant keeps two strategies running simultaneously: the open, symmetrical, beautifully arranged visible flowers that invite the world in, and the quiet, hidden, self-sufficient closed flowers that need no one and nothing to carry out their work. The violet is, in this way, a kind of philosopher, extravagant and social in fair weather, private and self-contained when the world withdraws. Many other plants have cleistogamous flowers or something similar. Some grasses flower in ways that are technically visible but never actually open wide enough to admit a pollinator, self-fertilizing in the sheltered space within their own husks. Some violets produce hundreds of times more seeds from their hidden flowers than from their visible ones. The mathematics of the visible flower, the bilateral symmetry, the nectar guides, the landing platform arranged for a specific pollinator, is only one strategy among several that plants have developed for the same fundamental purpose. The beauty is real, and it is also functional. And the function is served sometimes by forms that are not beautiful at all in a conventional sense, by the hidden, the closed, the self-sufficient. Let us rest a moment with the lily. The lily is one of the oldest flowers in cultivation. Lilies appear in ancient Egyptian artwork, in the ruins of Minoan palaces, in Chinese classical poetry, in the Song of Solomon. Humans have been growing and painting and writing about lilies for at least 4,000 years. And the lily is a beautifully, simply symmetrical flower. 6 petals arranged in 2 whorls of 3, with perfect radial symmetry, the petals alternating like the spokes of 2 overlapping wheels. The number 6 is not a Fibonacci number. It falls between the Fibonacci numbers 5 and 8, but it arises naturally from the lily's developmental biology. Lilies belong to a group of flowering plants called monocots, plants that produce one seed leaf when they germinate. Monocots tend to produce flower parts in multiples of 3 petals, or 6 or 9. Daffodils have 6 petals. Tulips have 6. Irises have their parts in 3s, 3 upright petals and 3 drooping ones. 3 is the monocot number, the way 5 is the number of the roses and the buttercups. This distinction between monocots and dicots, the 2 great divisions of flowering plants, is one of the deeper patterns underlying flower symmetry. Dicots, plants that produce 2 seed leaves, tend to have their flower parts in 4s or 5s. Roses, apples, geraniums, daisies, clover, all dicots, all tending toward petals in 4s or 5s. And the pattern holds remarkably well across the enormous diversity of flowering plant species. It is one of the first things a botanist learns to look for when identifying an unknown flower. Count the petals. 3 or 6, probably a monocot. 4 or 5, probably a dicot. The symmetry of the flower carries, encoded within it, information about the fundamental biology of the plant, about the very first moments of its life, when it was nothing but a seed in the soil, deciding how many seed leaves to unfurl. This is a thought worth sitting with very gently. The idea that the number of petals on a flower carries all the way back to the first moment of germination. That the symmetry of a bloom visible to anyone walking in a garden is an echo of a decision made in the dark underground when the seed first began to become a plant. The flower is a record of its own beginning. Every lily's 6 petals whispers something about how it started. Every rose's 5 petals is a kind of memory. And if you are finding these slow thoughts a comfort, if your breath is easier and your body is heavier in the good, restful way, please do share this with someone else who might need a quiet night. Or leave a little comment so we know you were here. This channel is built one gentle evening at a time, and knowing you're listening makes all the difference. Now let us think about what happens when symmetry breaks, because not everything in the flower world is perfectly symmetrical. Some flowers carry what botanists call floral asymmetry, and these asymmetries are themselves full of meaning. The twisting orchid is a good example. Most orchid flowers are bilaterally symmetrical, and they arrive in the bud with a specific orientation. But as the bud develops and ripens, the flower's stalk, called the pedicle, rotates through 180 degrees, twisting the flower so that the petal, called the lip, which is the broad landing platform for pollinators, ends up at the bottom rather than the top. This rotation is so reliable in most orchid species that botanists have a name for orchids in which the rotation fails to happen and the lip ends up at the top. They call them resupinate orchids, and when one fails to rotate, botanists say it is non-resupinate. The asymmetry that looks at first glance like a violation of symmetry is in fact the result of a very precise and deliberate rotation, a symmetry of process, if not of static position. Other flowers are asymmetric because of the way they are arranged on their stem. A flower that faces straight outward from a stem might be radially symmetrical when looked at head-on, but the way it is attached to the stem makes the whole structure, flower plus stem, bilaterally symmetrical, because the stem provides an axis, a reference point, a direction. And so, even flowers that are, in themselves, radially symmetrical become part of a larger bilaterally symmetrical arrangement when you consider them in their context, in relation to the stem that carries them and the leaves that surround them and the direction from which light falls on them. The broader lesson, if we are drawing one very softly, is that symmetry in flowers is not a static property, not a fixed characteristic that a flower either has or doesn't. It is dynamic. It is contextual. It is something that changes as the flower develops, as the bud opens, as the light changes, as you move around the flower and see it from different angles. The symmetry of a flower is a conversation between the flower and the world around it, between the flower's internal mathematics and the external influences that shape how that mathematics is expressed, which is, perhaps, a way of saying that symmetry is alive. Let us spend some time now with the most ancient flowers. Flowers are a relatively recent invention in the long history of life on Earth. The first flowering plants, the angiosperms, appeared somewhere between 130 and 140 million years ago, in the early Cretaceous period, at a time when dinosaurs were still the dominant large animals on land, and the continents looked very different from the way they look today. Before flowers, the world was full of gymnosperms, cone-bearing plants like conifers and cycads and the ancient ginkgo tree, and ferns and mosses and other plants that reproduced without flowers at all. The arrival of the flower was one of the most significant events in the history of life. because flowers changed the way plants and animals related to each other. They made a new kind of partnership possible, the partnership of pollination, the exchange of pollen for nectar, the coevolution of plant and animal that eventually produced, among other things, the extraordinary bilateral symmetry of the orchid and the hawk moth's matching tongue. The earliest flowers were, scientists believe, radially symmetrical. Bilateral symmetry evolved later, multiple times, independently, in different lineages of flowering plants. The orchids developing it for their purposes, the legumes developing it for theirs, the snapdragons for theirs. Each time bilateral symmetry evolved, it seems to have done so in response to a specific pollinator or a specific set of ecological conditions. And each time, it conferred an advantage because a flower that is more precisely matched to its pollinator gets more reliably pollinated, produces more seeds, has more descendants. Bilateral symmetry in flowers is a product of a very ancient and very patient process of fitting together, of mutual adaptation, of two living things adjusting to each other across geological time. There is a scientist named Daniel Jansen who spent many years studying what he called ecological fitting, the way that species in the modern world often show adaptations to partners that no longer exist, partners that went extinct thousands or millions of years ago. There are fruits in the Americas, for example, very large and very sweet fruits that seem designed for a large animal to eat and disperse their seeds. But the animals that ate those fruits, the giant ground sloths, the mastodons, the Pleistocene megafauna, went extinct 10,000 years ago. The fruits are still there, still producing their large, sweet, seemingly over-engineered flesh, still shaped for a partnership that no longer exists. And scientists have found something similar in certain flower-pollinator relationships. A flower whose symmetry seems designed for a specific insect or bird or bat that has since disappeared from its range. The flower still grows. The symmetry is still precise. It still carries the mathematical imprint of a relationship, a conversation with a partner that has gone quiet. Let your eyes close a little more if they haven't already. Let the muscles of your face be soft. There is nothing to see right now that requires looking. We are just thinking very gently about ancient flowers and ancient conversations, and the way the shapes of things carry the memory of the relationships that shape them. We should speak now about the geometry of the flower from the perspective of someone very small. A bee approaching a flower is navigating by symmetry. Experiments in which researchers created artificial flowers with different levels of symmetry and presented them to bees have shown that bees strongly prefer symmetrical flowers to asymmetrical ones, and that this preference is present even when the bees cannot see the flower directly, but only its shadow. The bees are detecting symmetry as a signal of quality because, in the natural world, a highly symmetrical flower is usually a healthy flower, a flower that has grown without nutritional stress, without significant disease or parasite damage, without the disruptions that can cause developmental asymmetries, will tend to be more symmetrical than a flower that has struggled. The bee, reading the symmetry of a flower, is reading a report on the flower's health, its resources, the quality of its nectar and pollen. The geometry is information, and the bees are extraordinarily sensitive to this. Researchers have found that bees can detect asymmetries in flowers that are too small to be detected by the human visual system. Differences of a degree or two in the angles between petals, differences in the width of petals measured in fractions of a millimeter. The bee's brain processes symmetry with a speed and precision that our brains can barely match, even with conscious effort and measuring instruments. For the bee, symmetry detection is automatic, immediate, effortless. The way reading text is for a fluent reader, automatic and effortless, and very fast. And the flowers, in response, have evolved toward higher and higher levels of symmetry because the bees reward it. A slightly more symmetrical flower gets visited more often, gets more reliably pollinated, produces more seeds. Over thousands of generations, the flowers that happen to be a little more symmetrical have more descendants, and those descendants tend to be a little more symmetrical too. And the process continues, generation after generation, the bees pulling the flowers toward ever more precise geometry with every visit, every preference, every moment of hovering in front of one flower rather than another. The extraordinary symmetry of a modern rose or orchid or lily is, in part, the accumulated aesthetic judgment of millions of generations of bees, which means that the beauty of flowers is not purely a human perception. It is a perception built into the sensory systems of pollinators who were refining their sense of floral beauty hundreds of millions of years before the first human being looked at a flower and felt moved. We arrived late to this appreciation. The bees were here first. And isn't that a soft and wonderful thought? To know that we share our love of beautiful flowers with the bees, that the same quality we find delightful in a garden, the same symmetry and balance and geometric grace, is what the bees have been choosing all along with their tiny and exquisitely sensitive eyes. If you haven't yet explored our playlist of sleepy science journeys, there is a whole world of these gentle stories waiting for you. From the mathematics of snowflakes to the science of why the sky turns pink at sunset. Each one is made for exactly this, for quiet evenings, for the slow drift toward sleep, for the pleasure of learning something beautiful without having to try too hard. You can find it just below the video, and we would love to have you there. Let us think now about the color patterns that develop within the petals of a single flower, the way a pansy has a dark center and lighter outer regions, the way a tiger lily is spotted, the way a tulip can be one color at its base and entirely different at its tip. These patterns are, again, arranged with mathematical precision. The spots on a tiger lily, the dark, almost black spots scattered across the orange petals, are arranged in a pattern that reflects the underlying symmetry of the petal. They are not random. They appear at specific positions at specific intervals, in a distribution that mirrors the left-right axis of the petal. Two spots on the left of the central vein are matched by two spots at the same position on the right. The pattern is bilateral at the level of individual spots. This spotting arises through a mechanism called reaction diffusion, first described mathematically by Alan Turing, the same Alan Turing who helped break the Enigma code during the Second World War and who laid the theoretical foundations for modern computing. In 1952, near the end of his life, Turing published a paper called The Chemical Basis of Morphogenesis, in which he described how two chemicals diffusing through tissue at different rates, could interact to produce regular patterns, spots, stripes, rings, spirals. He showed, purely mathematically, that very simple interactions between just two kinds of molecules could generate the complex patterns we see on animal skins and, as later researchers discovered, in flower petals. The spots on a tiger lily, the stripes on a zebra, the rings on a sand dollar, all of them potentially arising through the same class of mathematical mechanism, first described by a man who spent his career working on some of the most abstract and powerful mathematics of the 20th century. Turing did not live to see his ideas about biological pattern formation fully validated. That came decades later. But the patterns he described are real, and they appear throughout the living world, in flowers and animals and fungi and microorganisms, wherever chemistry meets growth and produces something regular and repeating out of something that might easily have been random. The symmetry of a flower's markings is, in this sense, a kind of spontaneous computation, a problem worked out cell by cell, molecule by molecule, in the slow time of development. And now we come, very gently, toward the end of our journey through the symmetry hidden in flowers. Let us spend these last quiet moments with a few final thoughts. Not big thoughts, not thoughts that require anything of you. Just small and soft observations, like the last few petals falling from a blossom at the end of its season. The petals of a flower fall in a particular way, not randomly, not all at once. They fall one by one, or two by two, often in a sequence that preserves the symmetry of the flower until the very end. Opposite petals falling at similar times. The bilateral or radial arrangement maintained even in the flower's dissolution. A rose losing its petals loses them in a spiral sequence that mirrors the spiral in which they first grew. The flower is symmetrical in its opening and in its closing, and in the slow, beautiful process of its ending. In Japan, there is a concept called mono no aware, the awareness of impermanence, the bittersweet appreciation of things precisely because they do not last. The Japanese tradition of hanami, the viewing of cherry blossoms, is built around this concept. People gather under cherry trees not simply to see the flowers at their peak, but to watch them fall, to be present for the brief, symmetrical snow of pink and white petals that fills the air for just a few days each year before the trees stand bare again. The symmetry of the falling petals, the way they spiral gently down from the branches in the light spring wind, is part of the beauty. The mathematics of the spiral persists even after the flower is gone, in the path of each petal through the air. Many cultures have used flowers as symbols precisely because of this combination of beauty and impermanence. Because a flower is something whose perfection is tied to its transience, whose geometry is most vivid when you know it will not last. The Fibonacci numbers in its petals, the golden angle of its arrangement, the bilateral symmetry of its form, all of this is expressed in the world for a brief time, in a particular light, for a particular set of eyes, and then it is over. But the mathematics persists. The next flower that opens on the same plant will follow the same sequence, the same angles, the same proportions. The pattern is deeper than any individual blossom. It lives in the instructions the plant carries within itself, in the chemistry of its growing cells, in the ancient conversation between the plant and its pollinators that has been going on for 100 million years. You are carrying, right now, in your body, the products of a similar kind of ancient patient process. Your own symmetry, the bilateral symmetry of your face and your body, the paired organs that mirror each other across your midline, the slightly imperfect but deeply present balance of left and right that makes you recognizably yourself. We are symmetrical creatures living in a world of symmetrical flowers, all of us shaped by the same underlying mathematics, all of us reading the same patterns in the world around us because we were shaped by the same world that shaped the flowers. The bee's love of floral symmetry and our love of floral symmetry are perhaps not so different. Both of them responses to the same deep order in the world, the same quiet mathematics running through living things, the same ancient geometry expressing itself in petals and wings and the curves of faces in the light. Let that thought be very light, like a petal on water. You don't have to hold it. You don't have to understand it fully or remember it. Just let it be there, floating. Let us return now to the question of what a flower knows about time. Not in any mysterious or mystical sense, but in the very real and measurable sense that flowers are tuned to time of day, to the time of year, to the rhythm of light and dark that moves through every 24 hours like a tide. Many flowers open and close on a precise schedule, and that schedule is governed by an internal clock, a biological clock made of proteins and chemical reactions that keeps time with remarkable accuracy even in the absence of external cues. If you were to bring a flower into a room with constant, unchanging light, it would still open and close on roughly its usual schedule, following the rhythm its cells have been keeping for generations. The clock is inside. The schedule is internal. The flower carries time within itself the way it carries the Fibonacci sequence within itself, not as something learned, but as something built. Carl Linnaeus, the Swedish botanist who gave us the system of Latin names we still use for all living things, was so charmed by this quality of flowers that in the 1750s he designed what he called the flower clock, Horologium Floriae in Latin. He had observed that different species of flowers opened and closed at different times of day with considerable consistency. He proposed planting a garden arranged in a circle, each sector planted with a species that opened or closed at a different hour, so that a walk around the garden would tell you the time. The dandelion opens at 5 or 6 in the morning. The African daisy opens at 7. The scarlet pimpernel opens at 8. The gazania opens at 10. And so on through the afternoon and evening, different flowers taking their turns at openness, each one reading its internal clock and responding. Linnaeus believed that a properly arranged flower clock could tell time to within half an hour. This flower clock was never built as a practical timepiece. It had obvious limitations, since clouds, temperature, and other variables affect flowering times. But the concept captures something true about flowers and time. Each species has its own schedule, its own preferred hours of openness, shaped by the schedules of its preferred pollinators. A flower that is visited primarily by bees, which are most active in mid-morning, opens in mid-morning. A flower visited by hummingbirds opens through the long afternoon. A flower visited by moths opens as the light fails, at dusk or in the early dark. The timing of a flower's opening is as much a part of its relationship with its pollinators as the shape of its petals or the arrangement of its symmetry. And the precision of that timing, the fact that it is driven by an internal clock that runs independently of external conditions, means that the flower can be ready and waiting at the right moment, even in variable weather, even on a cloudy morning when the light cues are ambiguous. The internal clock of a flower is made of proteins that interact with each other in a cycle. One protein promotes the production of another, which eventually inhibits the first, which allows the first to accumulate again, which starts the cycle over. The cycle takes roughly 24 hours, hence the name circadian, from the Latin for approximately a day. These circadian rhythms are found not just in flowers, but in almost every living thing on Earth. In fungi, in insects, in fish, in mammals, in human beings. The same basic molecular mechanism, proteins turning each other on and off in a cycle, appears to have arisen very early in the history of life and to have been preserved ever since. Because keeping track of the 24-hour cycle of light and dark is fundamental to surviving in a world that rotates. The flower that opens at the wrong time of day, that presents itself when its pollinators are absent or asleep, is a flower that gets visited less and produces fewer seeds. And so the clock was refined over millions of years until it ran with the accuracy that Linnaeus was moved to build a garden around. Imagine a field of wild flowers in the very early morning, before the sun has fully risen, the light just beginning to define the shapes of things in gray and pale gold. The dandelions are already open. You can see their yellow faces turned toward the east, waiting. The daisies are still closed, their petals folded up around their yellow centers, still an hour or two from their appointed time. The evening primroses from the night before are beginning to close, their pale yellow petals slowly curling inward as the light intensifies. Each flower on its own schedule, each one keeping its own time, the whole field a kind of slow and fragrant clock, turning and opening and closing in a sequence as precise and as beautiful as the spirals of seeds in a sunflower's face. You can let this image be very quiet and very still in your mind. The light is soft, the air is cool, the flowers are keeping time. Let us think now about fragrance, because fragrance and symmetry are, as it turns out, connected in ways that took scientists a long time to notice. It has been found in a number of flower species that the more symmetrical a flower is, the more intensely fragrant it tends to be. This is not a coincidence. Both symmetry and fragrance are indicators of floral quality. A healthy, well-nourished plant tends to be more symmetrical and to produce more volatile compounds, the molecules that evaporate from petals and travel through the air to reach the senses of pollinators. Both symmetry and scent are, in a sense, honest signals of a flower's condition. A flower cannot fake a high degree of symmetry without having the resources to maintain precise developmental control. It cannot produce large quantities of fragrant molecules without having the metabolic resources to spare. So, a flower that is highly symmetrical and strongly fragrant is advertising, honestly, that it is a high-quality flower with abundant nectar and pollen. And pollinators, over millions of years of evolution, have learned to read both signals. The molecules that make up floral fragrance are extraordinarily diverse. A rose's scent is produced by over 400 distinct chemical compounds, each one contributing something slightly different. A sweetness here, a hint of green there, a depth that comes from molecules too heavy to evaporate quickly, but present nonetheless. Jasmine, which smells similar to many people, actually shares very few of its volatile compounds with rose. Each flower species has its own molecular signature, its own particular blend of compounds, tuned to attract its particular pollinators. A flower visited by bees produces compounds that bees find appealing. A flower visited by flies may produce compounds that mimic the smell of meat or dung. A deceitful strategy, common in certain species, by which the flower lures pollinators with false promises rather than true rewards. But these deceitful flowers are the exception. Most floral fragrance is honest. Most floral fragrance is a true report of what the flower has to offer. The molecules of fragrance travel in a plume that drifts downwind from the flower, gradually dispersing as they mix with the air, growing more dilute with distance. A bee approaching a flower through a garden is navigating through a three-dimensional landscape of scent concentrations, moving toward regions of higher concentration the way you might move toward a fire on a cold night, following the warmth. The bee's antennae are extraordinarily sensitive to these molecules. They can detect concentrations far below what we could smell in quantities measured in parts per trillion. And the scent of the flower, combined with its visual symmetry, guides the bee toward the landing where the nectar and pollen wait. The symmetry you see and the fragrance you smell are, together, a single advertisement, a single message, a single offering. And the bee reads both simultaneously, integrating them into a single experience of approach and arrival. There is a flower called the bee orchid, and it is one of the most extraordinary examples of floral mimicry in the world. The bee orchid, which grows in meadows and chalk grassland in Europe and parts of Asia, has a lip petal, the broad landing platform of the orchid flower, that is shaped and colored to resemble, in remarkable detail, the body of a female bee. The brown, furry textured lip mimics the abdomen of a bee. The other petals mimic the wings. The flower even produces chemical compounds that mimic the pheromones, the scent signals, that female bees of a specific species release when they are ready to mate. A male bee, encountering a bee orchid, is deceived by the combination of visual and chemical mimicry into attempting to mate with the flower. In the process of this attempted mating, the bee picks up the orchid's pollen and carries it to the next bee orchid it encounters, where the same process occurs, and the pollen is transferred. The bee orchid is bilaterally symmetrical, like all orchids, but the symmetry serves a more complex purpose here. The lip must not only be symmetrical to look like a bee's body, it must be bilaterally symmetrical in the same way that a bee's body is bilaterally symmetrical, with the same axes of mirror reflection, the same proportions, because a male bee recognizing a potential mate is doing so through a perceptual system evolved to assess exactly those symmetries. The orchid is exploiting the bee's own symmetry detection system, using the bee's evolved preference for bilaterally symmetrical forms to engineer its own pollination. The flower has become, through millions of years of selection, a kind of three-dimensional painting of a bee, a painting accurate enough to fool, at close range, the very creature it is portraying. It is worth pausing with the extraordinary improbability of this, that a flower could come to resemble, closely enough to deceive, the body of an insect. That the same mathematical processes of differential growth that produced the gentle cup of a rose petal could produce, in a different species, a structure that mimics fur, that captures the visual bilateral symmetry of a bee's abdomen, that even produces the right chemical compounds to complete the illusion. The bee orchid is a reminder that the mathematics of flower development is capable of generating not just beauty, but complexity. Not just patterns that are pleasing to our eyes, but structures precise enough to deceive the sophisticated perceptual systems of another living creature. And if you are still here with us, still drifting through this garden of slow science, thank you for being here. Do check out the playlist if you haven't yet. There is a whole night's worth of gentle science waiting for you there. And each video is made with exactly this in mind: your rest, your quiet, your slow drift toward sleep. Let us rest now with the mathematics of petals and time together, with the way a flower opens. The opening of a flower bud is one of the slowest and most beautiful processes in the natural world, and it is driven again by differential growth. When a bud is ready to open, the cells on the inner surface of the petals, the surface facing the inside of the bud, begin to grow faster than the cells on the outer surface. Because the inner surface is expanding and the outer surface is not, the petal curls outward. One by one, or all together, the petals uncurl from the center of the bud, each one rotating outward as its inner cells outgrow its outer cells, until the flower is fully open and the petals lie more or less flat, or are slightly reflexed, curled back, depending on the species. The mathematics of this opening is the same mathematics as the curling of a heated bimetallic strip, the same as the curling of a drying autumn leaf. A structure that is constrained on one side and not the other will curl toward the constrained side. Some flowers open in seconds or minutes. The morning glory, an appropriate name, opens in the very early morning, its trumpet-shaped blue or purple flowers unfurling from their spirally furled buds in a process visible to the naked eye if you watch closely enough. Time-lapse photography of flowers opening reveals this as a kind of dance. The sepals first pushing aside, then the petals unfolding, each one following the last in a sequence determined by the geometry of the bud, until the full flower is revealed. In the reverse direction, the closing of a flower at the end of the day. The same mathematics runs in reverse. The outer cells of the petals now outgrowing the inner cells, curling the petals back inward and shut. The same flower, the same petals, the same cells, producing opposite movements at opposite times of day, driven by the flower's internal clock and the direction of that clock's signal to the growing tissue. There are flowers that open so quickly that the movement is visible to the naked eye without any time lapse at all. The white water lily opens its flowers in a matter of minutes, the petals spreading smoothly outward from a tight bud in a motion that looks deliberate and conscious, though it is purely chemical and mechanical. And there are flowers that open extremely slowly. Some tropical orchids take days to fully open from the bud, the petals moving so gradually that no individual moment of movement can be detected. The flower that opens in minutes and the flower that takes days are using the same biological mechanism. They are simply running it at different speeds, turning the dial of differential growth up or down according to the instructions encoded in their cells. There is a kind of symmetry in flowers that we have not yet spoken about, a symmetry across time. Not the spatial symmetry of petals and spirals and golden angles, but the symmetry between the flower and every other flower of its species, across all the time that species has existed. When a buttercup opens its 5 petals in a meadow this spring, it is expressing a form that has been expressed, more or less unchanged, by buttercups for millions of years. Fossils of early buttercup relatives have been found that show the same radial arrangement, the same Fibonacci petal counts, the same basic floral geometry as the flowers we see today. The form has been stable across geological time, across ice ages and warming periods, across the extinctions of many of the buttercup's original pollinators and the arrival of new ones, across continental drift and the slow rearrangement of the world's landscapes. This temporal symmetry, the way each new flower echoes all the flowers that came before it, is possible because the instructions for making the flower are encoded in the plant's DNA, and DNA is extraordinarily good at being copied faithfully from one generation to the next. Each buttercup seed carries within it the complete instructions for making a buttercup flower. The same golden angle, the same number of petals, the same arrangement of stamens and pistille, the same chemical compounds for fragrance and nectar, and those instructions have been copied with very high fidelity from parent plant to seed to new plant for millions of generations. The form persists because the instructions persist. The geometry of the flower is a kind of memory, a molecular memory, encoded in DNA, faithfully reproduced, flowing forward through time in an unbroken chain of copies. Of course, the copies are not perfectly faithful. There are occasional errors in copying, mutations, and sometimes those errors produce a slightly different flower, a petal that is a slightly different shade, a stamen that is slightly longer, a fragrance molecule that is slightly different. Most of these errors make no difference or make the flower slightly less fit. But occasionally, very occasionally, an error produces something useful, a flower that is slightly more attractive to its pollinators, or slightly more resistant to a fungal infection, or slightly better at opening at the right time of day. Those errors persist and spread because the plants that carry them produce more seeds. And over millions of years, the accumulation of useful errors, each one a tiny deviation from the previous form, has produced the extraordinary diversity of flowers we see in the world today. The 25,000 species of orchid, each one different, each one exquisitely fitted to its particular corner of the world, all of them descended from some ancient flower ancestor whose form has been varied and refined and diversified across the whole long history of plant life on Earth. The diversity is the result of accumulated deviation. The stability is the result of faithful copying. Both are happening at the same time in every generation, in every flower. The sameness and the difference, the repetition and the variation, the symmetry and the asymmetry, balanced against each other in an ongoing negotiation between the pressure to copy faithfully and the pressure to adapt and change. It is the same negotiation that produces the diversity of life in general, the same balance between conservation and innovation, between the old form and the new possibility. Let these thoughts be very light. Let them float. You don't need to hold any of them. They are just the soft science of flowers drifting through your mind on a quiet evening, on their way to wherever thoughts go when we sleep. We have spoken about flowers that deceive, the bee orchid, the fly orchid, the various species that produce smells of meat or rotting matter to lure carrion flies into pollinating them. But let us spend a moment with a different kind of deception, one that is gentler and more whimsical, the false petal. Many plants that appear to have large, colorful petals are in fact displaying something that is not a petal at all, a modified leaf called a bract that has taken on the color and shape of a petal in order to attract pollinators to the true, often very small flowers at its center. The poinsettia, which we associate with winter and warmth and the particular red of Christmas decorations, has tiny yellow flowers clustered at its center. The enormous red structures that we think of as the poinsettia's petals are not petals at all. They are bracts, modified leaves, transformed by evolution into petal mimics so effective that most people never question what they are looking at. The dogwood tree blooms in spring with what appear to be 4 large white petals surrounding a small cluster of flowers. The 4 white structures are bracts, each one a modified leaf that has become white and petal-shaped and arranged with the radial symmetry of a petal to serve the same function as a petal, attracting pollinators from a distance. The real flowers of the dogwood are small and modest, clustered at the center, doing the actual work of reproduction. But the bracts do the advertising, the color, the display, the symmetrical arrangement that catches the eye of a bee or a butterfly from across a garden. The bougainvillea, with its astonishing explosions of color, magenta, orange, white, red, is similarly deceptive. The true flowers of the bougainvillea are small and white, almost inconspicuous, held in groups of 3. The color comes from 3 papery bracts surrounding each group of 3 flowers, each bract taking the approximate shape of a petal, and the whole arrangement repeating in masses along the vine until the effect is overwhelming. What you see when you look at a flowering bougainvillea is an architecture of illusion. Thousands of small, careful, 3-part deceits, each one a set of 3 petal mimics surrounding 3 true flowers, all of it adding up to something that registers to human eyes and pollinator eyes alike as an abundance of flowers, an extravagance of color, a generous and beautiful display. It is a kind of theater performed in slow biological time, the stage set built out of leaves. Now let us think about the smallest flowers. The smallest flowers in the world are the flowers of a plant called Wolffia, a genus of aquatic plants sometimes called watermeal or duckweed, though true duckweed is a slightly different plant. Wolffia is extraordinary for several reasons. First, the plants themselves are extraordinarily small. A Wolffia plant is a tiny green oval, roughly the size of a grain of salt, floating on the surface of still water. Second, these tiny plants flower. They produce flowers so small that they are visible only under magnification. Flowers that consist of nothing more than a single stamen and a single pistil, without any petals at all. The flower is as reduced as a flower can be, stripped of all decoration, all color, all fragrance, all symmetry of petals, down to the bare essentials of reproduction, male and female parts in proximity. Wolffia reproduces primarily by vegetative reproduction, simply dividing and producing copies of itself, budding off new plants without any sexual reproduction at all. Its flowers are used occasionally, but the plant has largely abandoned the elaborate investment of resources that most flowers represent. No petals to grow, no nectar to produce, no fragrance to synthesize, no color to display. The reduction is complete. The Wolfia flower is a flower at its minimum, a single fact expressed in living tissue. And yet, even the Wolfia flower has symmetry. The single stamen and single pistil are arranged in a radially symmetrical relationship to each other. The tiny flower's opening, barely visible to the naked eye, is as precisely structured as a rose, just with almost nothing in it. The mathematics of flower development, the same developmental rules that produce the elaborate spirals of a sunflower or the bilateral symmetry of an orchid, produces in Wolfia a form so simple that it is almost not there. The flower is a whisper of what a flower can be. It is the minimum expression of the same deep biological program that, in other circumstances, becomes a rose. There is something very calming about this, about knowing that the same instructions can produce both the rose and the Wolfia flower, both the enormous, extravagant complexity of an orchid and the single stamen simplicity of the world's smallest flower. The program is not fixed at a particular level of complexity. It can be turned up or turned down, elaborated or reduced across a vast range of expression, while remaining at its heart the same program, the same Fibonacci mathematics, the same circadian timing, the same differential growth, expressed more or less fully depending on the ecological and evolutionary pressures each species has faced. We should speak now about the geometry of the flower from above. The aerial view, the view that a bee has as it descends toward a landing. From directly above, a radially symmetrical flower looks like a target, a central disc surrounded by evenly spaced petals, the whole arrangement radiating outward from the center with perfect evenness. And this target quality is again intentional. The radial symmetry of the flower, seen from above, functions as a visual bullseye, drawing the eye and the bee's compound eye toward the center where the nectar and pollen are. The bee does not need to search for the center. The geometry of the flower makes the center obvious. Compound eyes, like those of bees and other insects, perceive the world differently from the simple eyes of vertebrates like us. A compound eye is made up of hundreds or thousands of individual light-detecting units called ommatidia, each one pointing in a slightly different direction, giving the insect an extremely wide field of view, but with lower resolution than our eyes provide. A bee's eye sees a flower not as a sharp, detailed image, but as a pattern of light and dark and color, a kind of pixelated impression. And yet bees navigate toward flowers with extraordinary accuracy, landing precisely on the structures they intend to land on. The radial symmetry of flowers is, in part, adapted to the visual system of the bee. It is a pattern that reads clearly, even at low resolution, as a target centered on a point. The flower's geometry is calibrated for the eye that reads it. Experiments in which researchers created artificial flowers with different levels of radial symmetry, some perfectly symmetrical, some with slightly uneven petal spacing, some with very uneven spacing, and presented them to bees, found that bees could detect and preferred symmetry even when the deviations were very small, smaller than the resolution of a bee's visual system would theoretically allow. This suggests that bees are detecting symmetry not just visually, but through some combination of visual and other cues. Perhaps the symmetrical arrangement of the flower's scent glands, producing an equally symmetrical scent plume, reinforces the visual information. The flower is symmetrical in every sensory channel simultaneously, and the bee is reading all of them at once, integrating them into a single, confident assessment. This is a high-quality flower worth landing on. And if this feels good to you, this slow descent through the science of flowers, this gentle accumulation of soft facts, do leave us a like. It takes a moment, and it means a great deal. And the playlist is always there when you need more of this quietness. Let us think finally in this part of our journey about what flowers do for each other, about the way they cooperate and compete, the way the presence of many flowers together in one place changes the mathematics of pollination for all of them. When many flowers of the same species bloom at the same time, a phenomenon called mass flowering, they collectively attract more pollinators than any single flower could, because a large display is visible from farther away and is more rewarding for pollinators who make the trip. But mass flowering also means that more of the pollen moving around is from the same species, which reduces the chance of cross-species contamination. And the timing of mass flowering is itself governed by the same circadian and seasonal clocks we have already discussed, by day length, by temperature, by the accumulated warmth of spring, the symmetry of a mass flowering event. A cherry tree in full bloom, every branch covered in blossoms, every blossom opening within a few days of every other, is a kind of superorganism symmetry, the symmetry of the whole tree expressing itself in coordinated time, thousands of individual flowers all following the same clock, all responding to the same signal, all opening together in a display that is, collectively, far more than any single flower. And the cherry tree's timing is not independent of other cherry trees. Trees respond to many of the same environmental signals, and cherry trees in the same location tend to flower at the same time, creating the wave of blossoms that moves across a hillside or a garden in the days of early spring. The flowers of one tree echoing the flowers of its neighbors, the symmetry spreading outward through the landscape. The word for the time of peak cherry blossom in Japanese is mankai, full bloom, full flowering. It is a word with a quality of completion about it, of a thing being fully what it is. And the mankai of a cherry tree, the moment of perfect symmetry between the tree's internal time and the external conditions of spring, lasts only a few days before the petals begin to fall. It is a precision of timing as exact as the golden angle in a sunflower, as the bilateral symmetry of an orchid, a mathematical exactness expressed not in space, but in time, in the perfect moment of full, open, radiant bloom. You can imagine it now if you like. A hillside of cherry trees, all in mankai, all open at once, the air full of petals in the light. And then, very slowly, the petals beginning to drift, the mathematics of the flower relaxing after all its precision into the gentle randomness of falling. Let us spend some time in the deep history of flowers one more time, because there is a story there about how symmetry itself evolved that is worth telling slowly and softly. When the first flowering plants appeared, about 130 to 140 million years ago, they had to compete with the established gymnosperms, the conifers, cycads, and other cone-bearing plants that had dominated the land for hundreds of millions of years. The early flowering plants were small and modest, probably weedy in the ecological sense, fast-growing opportunists, filling disturbed areas, producing their seeds quickly. But they had one new thing, the flower, and the flower gave them a new kind of relationship with the animal world. The earliest flowers were probably simple, radially symmetrical, with many petals and many stamens, something like a modern magnolia blossom or a water lily. They were probably pollinated by beetles. Beetles were the most abundant insects of the Cretaceous period, and beetle-pollinated flowers tend to be open, bowl-shaped, radially symmetrical, without the precision engineering of flowers that target specific bees or moths. The earliest flowers, in other words, were generalists. They accepted whatever pollinator came their way. They had no particular partnership, no elaborate bilateral symmetry tuned to a specific tongue length or body size. They offered pollen as a food reward, and the beetles came for the pollen and accidentally transferred some from flower to flower, and this was enough. But over time, as new groups of insects evolved, particularly the bees, which appeared about 130 million years ago, and the butterflies and moths, which appeared somewhat later, the possibility of more specific relationships emerged. A flower that happened to be slightly more attractive to a particular kind of bee. A flower that happened to present its pollen at an angle that deposited it on exactly the right part of a specific bee's body. A flower that happened to have bilateral symmetry that matched a bee's preferred approach angle. Such a flower would be more reliably pollinated than its neighbors and would leave more descendants. And those descendants would carry the same slight advantages, slightly amplified. And so, over millions of years, from the simple generalist flower of the early Cretaceous, the entire extraordinary diversity of floral forms evolved, each one a specific answer to a specific question, each one a mathematical solution to the problem of getting pollen from one flower to another. The symmetry of flowers is, in this sense, a record of evolutionary history, a fossil of relationships, written in geometry rather than stone. A radially symmetrical flower speaks of a generalist strategy, an open invitation. A bilaterally symmetrical flower speaks of a specific partnership, a conversation conducted in angles and proportions between two species that have been shaping each other for a very long time. And the extraordinary diversity of floral symmetries in the world today speaks of the extraordinary diversity of those relationships, the vast branching network of partnerships between plants and animals that has been building relationship by relationship for 100 million years. There is something very peaceful about this, seen from the right distance, in the right light, at the right speed. The world, over enormous stretches of time, has been arranging itself into relationships, into partnerships, into the mutual fittings of form and function that we call co-evolution. The flower and the bee, the orchid and the moth, the poinsettia and the hummingbird, all of them part of an ancient ongoing process of the world coming to know itself through the relationships of its parts and the symmetry of every flower you have ever seen, every rose in a garden, every daisy in a field, every orchid in a window, is part of that enormous, quiet, beautiful process. Let that rest in your mind very lightly, like pollen drifting, like a petal on water. Now, let us think about what happens inside the flower after pollination, after the pollen has been carried from one flower to the next and has landed on the receptive surface of the pistil. This process, called fertilization, is invisible to us, hidden inside the flower, conducted in chemical language, but it is as mathematically precise as everything else about the flower. When a pollen grain lands on the stigma, the sticky top of the pistil, it begins to grow a long tube called the pollen tube that grows down through the pistil toward the ovary at its base, where the egg cells wait. The pollen tube grows through a chemical gradient, a concentration of signaling molecules that is highest near the egg cells and decreases with distance. The tube follows this gradient, navigating toward the highest concentration, the way a river navigates toward the sea by following the gradient of elevation. It is chemotaxis, movement toward a chemical signal, and it is one of the most fundamental navigational strategies in all of biology. The pollen tube grows at speeds that vary by species. Some tubes travel their entire route in a matter of hours. Others take days. In some plants, the tube can be several centimeters long. Inside the pollen tube travel 2 sperm cells, 2 male reproductive cells that will eventually fertilize the egg cells in the ovary. In flowering plants, fertilization is actually double. 1 sperm cell fertilizes the egg cell, producing the embryo that will become the new plant. The other sperm cell fuses with another cell in the ovary to produce what is called the endosperm, a nutrient-rich tissue that surrounds the embryo inside the seed and provides it with food for its first days of growth. This double fertilization is unique to flowering plants. No other group of plants does it. It is one of the characteristics that defines the angiosperms as a distinct and extraordinary lineage. The seed that results from this double fertilization carries within it the complete genetic blueprint for a new flower, a new instance of the same mathematical program, the same Fibonacci numbers, the same golden angle, the same circadian clock, the same differential growth rules. The seed is a compressed version of all the mathematics we have been discussing, held in a tiny, dormant packet of cells, waiting for the right conditions of moisture and temperature and light to unfurl. And when it does unfurl, when the seed germinates and the first shoot reaches upward and the first leaves spread, the program begins again. The same program in a new place, in a new individual, in a new conversation with its particular patch of soil and its particular local pollinators and its particular piece of sky. The flower is, in this sense, a kind of message, a message from a parent plant to a future plant, carried in a seed across space and time. And the message is the mathematics of the flower. The symmetry is the message. The golden angle is the message. The bilateral or radial form, the Fibonacci petal count, the circadian timing, the ultraviolet nectar guides, all of it encoded in the DNA of the seed, waiting to be expressed again, to bloom again, to open again in whatever light finds it. What a quiet and extraordinary thought to take with you into sleep, that every flower that has ever opened has carried within it a message from every flower that came before it, that the mathematics of a petal is a letter written in chemistry and passed down through millions of generations, that the symmetry you see in a garden is the accumulated handwriting of geological time. There is one more aspect of flower symmetry that we have not yet touched, and it is one of the most intimate and the most quietly remarkable. The symmetry of a flower is not just visible from the outside, it is also felt, in a way, by the pollinators that touch it, and even by the plant itself. Plants have a form of mechanosensitivity, sensitivity to physical touch and vibration, that scientists have only begun to understand in the last few decades. A plant that is touched or vibrated will respond chemically, producing different amounts of certain hormones, changing the pace of its development, sometimes even changing the expression of certain genes. Bees, when they land on a flower, do not simply sit passively collecting nectar and pollen. They often vibrate at specific frequencies. They buzz. And this buzzing, which is produced by their flight muscles and conducted through the flower's petals and stamens, has been found to cause the pollen to be released more efficiently. The vibration loosens the pollen from the anthers, the pollen-producing structures, and causes it to fall more freely onto the bee's body. This process is called buzz pollination or sonication, and it is used by about 9% of the world's flowering plant species, including tomatoes, blueberries, cranberries, and many wildflowers. Bumblebees are particularly good at buzz pollination, and some flowers appear to have evolved specifically to require it. Their anthers are shaped in a way that releases pollen only in response to the right frequency of vibration, which only certain bees can produce. The symmetry of a flower plays a role in buzz pollination too. The stamens of a radially symmetrical flower that relies on buzz pollination are arranged evenly around the center so that wherever a bee lands, it contacts stamens in the same configuration. The symmetry ensures that the bee's vibration is applied evenly, that the pollen release is even, that the bee picks up a symmetrically distributed coating of pollen that will be deposited symmetrically on the next flower's pistil. Even at the level of vibration and physical contact, the flower's symmetry is serving a function, ensuring consistency, ensuring that the mathematical precision of the reproductive process is maintained regardless of the slight variations in how and where the bee lands. And the flower, in turn, responds to the bee's touch. Researchers have found that some flowers, after being visited by a pollinator, change their chemistry in ways that make them temporarily less attractive to subsequent visitors, reducing nectar production, changing fragrance output, so that the next bee is encouraged to move on to an unvisited flower. The flower is, in a sense, broadcasting its status, visited or unvisited, fresh or spent. It is managing the flow of pollinators through the garden, optimizing the distribution of visits across all the flowers on a plant, ensuring that every flower gets pollinated, rather than all the bees concentrating on the first few they find. The flower is not passive. It is participating actively in the process, adjusting its signals in real time in response to what it experiences. Let us think quietly about what it means that all of this has been going on, all of this extraordinary complexity and precision in every garden and meadow and forest for 100 million years, without any of it being planned or intended or consciously designed. The golden angle was not chosen. The Fibonacci sequence was not decided upon. The bilateral symmetry of the orchid lip was not drawn up as a blueprint. All of it arose through the same simple process: variation, selection, inheritance, running over and over again, generation after generation, in populations of plants and animals, each one slightly different from the last, the useful differences being preserved and amplified, and the less useful ones fading away. This process is sometimes called blind, undirected. But from where we are sitting in this soft evening, with the mathematics of flowers laid out gently around us, it does not feel blind. It feels patient. It feels enormously, extraordinarily patient. The patience of geological time, of 100 million years of variation and selection, of every generation, of every flower contributing slightly to the refinement of the form. The result, the flower as it exists today, in all its mathematical precision and its biological complexity and its quiet beauty, is what patience produces when patience is given enough time. Human beings have been patient in this way too, in the cultivation of flowers. Over the last 10,000 years, since people began to deliberately grow and select and breed plants, they have participated in the same process of variation and selection, but consciously, intentionally, with preferences and purposes. The roses in modern gardens are the result of centuries of deliberate breeding, selecting for size, for color, for fragrance, for repeat flowering, for the number and arrangement of petals. Modern roses can have far more petals than their wild ancestors. Some cultivated roses have 100 or more petals, packed into a dense, spiraling mass that bears only a family resemblance to the simple 5-petaled wild rose. The Fibonacci numbers are still there, still present in the arrangement of those petals, but elaborated and multiplied, the same mathematics running through a much more complex expression. And the flowers people have chosen to cultivate and breed and elaborate across these 10,000 years are, without exception, highly symmetrical ones. People did not choose to breed asymmetrical flowers into even greater asymmetry. They chose to take symmetrical flowers and make them more so. More petals, more evenly arranged, more perfectly balanced around the center. The human aesthetic preference for symmetry has shaped the cultivated flora of the world, pulling it toward greater and greater degrees of geometric precision. The symmetry of a formal rose garden is, in part, a record of human preference, a preference as old as our capacity for preference, as ancient as the first person who picked a flower and felt something in looking at it. One of the most beautiful and least commonly known aspects of flower symmetry is what happens when symmetry fails, when, for some developmental reason, a flower produces a different number of petals than its species usually produces, or produces petals of unequal size, or develops an extra whorl of stamens. These variations, called floral mutants or floral abnormalities, are usually the result of mutations in the genes that control floral development, and they have taught scientists an enormous amount about how the normal symmetry of flowers is produced. There is a system of genes called the ABC model of flower development, one of the landmark discoveries of plant biology in the late 20th century. The ABC model describes how 3 classes of genes, A, B, and C, work together to specify the different organs of a flower. Genes alone produce sepals, the green leaf-like structures at the base of the flower. A and B genes together produce petals. B and C genes together produce stamens. C genes alone produce carpels, the female reproductive structures. This elegant genetic logic, in which overlapping combinations of 3 classes of genes produce 4 different organ types, has been found to hold, with variations, across a very wide range of flowering plant species. And when one of these genes is mutated, when A genes are silenced, or B genes are duplicated, or C genes are lost, the results are dramatic and illuminating. A flower with no functional A genes has no sepals and no petals. Instead, it has carpels and stamens from the outermost ring inward. A flower with no functional B genes produces sepals where petals should be, and carpels where stamens should be. A flower with duplicated B function genes can have a double whorl of petals. The same mutation appearing over and over again in cultivated plants that produces the doubled flowers of roses, camellias, and peonies. The extra petals of a cultivated rose are the result of a disruption in the ABC model. Stamens that would normally be stamens have been converted into petals because the balance of A, B, and C gene activity has been shifted. The mathematical precision of the normal flower is produced by the precise balance of these genetic signals. When the balance shifts, the form shifts, and the form that results tells us clearly what the normal balance was producing. The mutant flower, in other words, illuminates the normal flower. The exception reveals the rule. The asymmetry reveals the symmetry. And in revealing it, it shows us something remarkable, that the beautiful, balanced, golden angled geometry of a normal flower is the product of a small number of interacting genetic signals, not a large and complex set of individual instructions, but a simple combinatorial logic of 3 classes of genes, their interactions specifying the identities of petals and stamens and carpels with elegant economy. The flower is not complicated to produce. It is produced by simple rules, applied precisely. The mathematics is simple. The result is beautiful. And if you are still following this thought, still here in the soft evening with us, please do give the video a like. It costs nothing, and it helps this channel continue to grow, one quiet night at a time. Let us come now toward the very last of our facts about the symmetry hidden in flowers, and let us choose for this final approach a thought that is simple and quiet and very close to the experience of being a person in the world. Every person who has ever looked at a flower and felt something, felt warmth or tenderness or a kind of wordless pleasure, has been responding in part to its symmetry. This is not a reductive thing to say. It does not make the feeling smaller or less real. It makes it larger because it connects that individual feeling to 1 billion years of biology, to the evolution of symmetry detection in nervous systems, to the ancient partnership between flowers and animals, to the mathematics of growth and the chemistry of fragrance and the light of particular mornings in particular gardens in the long human past. When you look at a flower and feel something, you are participating in one of the oldest things that animals do. You are reading a signal that has been refined across 100 million years of partnership between plants and the creatures that visit them. Your nervous system is doing something that bee nervous systems do, that butterfly nervous systems do, that the nervous systems of the very first flower visiting beetles did 100 million years ago in the warm forests of the early Cretaceous. The symmetry of the flower reaches into you and produces a response that is, in its deepest origins, the same response that has guided pollinators towards flowers since flowers first existed. And at the same time, it is entirely yours. The particular moment in which you last saw a flower and felt moved by it is yours alone. The particular light, the particular fragrance, the particular quality of that morning or afternoon or evening, the particular state of your heart and mind in that moment. The mathematics and the biology and the 100 million years of evolutionary history are all there in the background, in the deep structure of the experience. But the experience itself is yours, as immediate and as personal and as unrepeatable as any other moment of your life. Both of these things are true at once. The flower is ancient and mathematical and evolved and perfectly calibrated by deep time. And the flower is here, now, in front of you, opening in its particular light on this particular morning, and it is beautiful, and you are feeling something, and that is enough. That is, in fact, everything. The night has grown very deep now. The meadow around us is very quiet. The flowers are closed, every one of them, from the tiny dandelion at the edge of the path to the tall foxglove leaning against the stone wall, all folded in, all dark, all resting. Their circadian clocks are running in the dark, keeping time, waiting for the particular moment of morning when each will open again. The Fibonacci spirals in the seed heads are patient. The golden angle is encoded in every new growth tip, waiting for morning to continue the spiral. The bee orchid's petal that looks like a bee is furled and still. The bilateral symmetry of the iris is tucked away until tomorrow. Everything is folded, resting, held in the dark in the geometry of its closed form. And you too can fold in now. You too can let the symmetry of your breathing settle into something even and quiet. You too can let the day's accumulation of thought and effort and motion settle down through you and out of you, leaving just the even, unhurried rhythm of rest. Your body knows how to do this. It has been doing it every night of your life. The circadian clock in your cells is telling you, as it tells the flowers, that this is the time. This is the time for the inner cells to slow their activity, for the temperature to drop a little, for the eyes to close and the muscles to release, and the mind to loosen its grip on the particular arrangements of the waking world. Let the mathematics go. Let the spirals and the Fibonacci numbers and the golden angles drift out through the edges of your attention and into the soft, dark beyond. You don't need to hold any of it. The flower will still be symmetrical in the morning. The bee will still know how to read it. The golden angle will still be 137.5 degrees when you wake. None of it needs your attention to continue. It all continues beautifully, endlessly, without anyone watching. The world is very old and very full of quiet patterns, and you are part of it, resting now, folded inward like a flower at the end of the day, held in the dark, patient, waiting for morning. Everything is soft, everything is still. The only sound is the sound of your breathing, slow and even, and the very distant sound of wind moving through the petals of flowers you cannot see, in gardens you will never visit, opening and closing and opening again, patiently, beautifully, forever. You are safe. You are warm. You are held. Sleep now. Thank you for spending this long, quiet evening with the Sleepy Science Channel. The playlist is always waiting for you. Each story another gentle door into the wonders of the world. Good night.